Hello, my name is Tyler Bobbitt, and my RISE project is titled Differential Mitochondrial Subtype Segregation as a Driver of Cell-Fate Decisions in Mammals. Been mentored by Dr. Jonathan Tilley and Dr. Dory Woods, and currently a third-year undergraduate here at Northeastern. So for a quick background of my project, what it is kind of an interest in mitochondrial heterogeneity in pre-implantation embryos. So recently, there's been increasing awareness in the biological community that mitochondria are not the same. They can be different sizes, they can perform different functions, they can be different polarities or have different membrane potentials. What we're interested in is seeing, are there significant differences in this heterogeneity or between mitochondrial breakdowns in embryos at different stages? So between the 1-cell embryo stage, 2-cell stage, 4-cell, and 8-cell stages. Also, what we're interested in is this mitochondrial heterogeneity maybe playing a role in furthering embryo development? So between 8-cell embryos, which only have one lineage, and blastocyst cells that have 2 distinct cellular lineages, does the mitochondrial heterogeneity maybe within one cell in a blastomere, or in general, maybe influence or drive this lineage specification? So for a general method breakdown for our first heterogeneity experiment, we're looking at 1-cell, 2-cell, 4-cell, and 8-cell embryos. We're isolating the mitochondria, and then using our FAMS technology in the Tilley Woods lab, we are analyzing these pooled mitochondria at a single mitochondrial level for size and membrane potential. Additionally, we're performing and validating a tracing method for mitochondria where we can lyse, sort mitochondria, fluorescently label them, and then inject them into a 2-cell embryo in, say, the subzonal space, and allow that embryo to develop into a blastocyst with the 2 lineages, and fluorescently image them to look for mitochondrial localization. Currently, for our results for the heterogeneity experiments, real briefly, we haven't been able to see anything significant both for the size and for the membrane potential breakdowns. For our tracing experiments, though, we've had pretty good success. On this video on the left is me actually injecting sorted and labeled mitochondria into a 2-cell embryo. And as you can see on the right, these injected mitochondria were uptaken by the cell. The embryo was able to develop to the blastocyst stage, and we were able to fluorescently label them. So we were pretty confident that we validated this technique. Given this, there's a couple future directions we can have. For the heterogeneity experiments, we're hoping to just get a few more replicates to see if we can tease apart any heterogeneity or like any breakdowns within the groups. And also for the tracing experiments, we're hoping to sort different mitochondrial subtypes. So say small mitochondria versus large mitochondria, and perform the tracing experiments to see are specific subtypes maybe localized in the blastocyst differently. Like large mitochondria going into the ICM or small to the trophectoderm or vice versa, and see are there any significant differences. Yeah, so that's a quick breakdown for my project that I've been working on for the past about year. And thank you guys for listening.